So there has been many new BCMA-directed therapies for multiple myeloma the later years. I'm going through the different types here. So next slide. And here we have the overview of all, and they are divided into three groups. It's the antibody-drug conjugates on the left, the bispecific antibodies in the middle, and CAR T cells on the right. And they are also showed here by what indication they have. So if it's more than one or more, two or more, three or more, or four or more prior lines of treatment. And there's also a color distinction here for whether it's approved in Europe or both Europe and FDA. So you can see that belantamab, the only antibody-drug conjugate that is approved in Europe from second line, while only the combination with bortezomib is approved in US, and that's from third line. Looking at bispecifics, there are three approved bispecifics directed towards BCMA, teclistamab, belantamab, and vimseltamab, and they are approved in fourth line in Europe and in fifth line in US. For the CAR Ts, it's very similar for Europe and US. Cilta-cel approved in second line, ide-cel approved in third line. So next slide. So here we look at how they work. What's their mode of action? It's quite different in the three different types of targeting BCMA. On the left, you see the antibody-drug conjugates. I can first say that these are all sort of based on the antibody type recognition of BCMA, so that's quite similar. But what they do is quite different. So on the left, antibody-drug conjugates, it's an antibody that recognizes the BCMA, but it brings with it a payload, an old-fashioned chemotherapy, actually, which is internalized into cells that have BCMA on the surface. And that payload kills the cell. In the middle, you see the bispecifics. They have two antibody targets. They both target BCMA, but also CD3 on the T cell. What happens then is that they bring the T cells, which can kill myeloma cells, into close connection with the myeloma cells, and that leads to activation of the T cells, leading to quite rapid killing of myeloma cells with this mechanism. The third one is the most sort of complex one. Here you harvest T cells from the patient. These T cells are then modified to express what we call a CAR T product, which is something that outside recognizes BCMA on the myeloma cell as the other drugs here, but on the inside it activates the T cells directly. And also leading to a very efficient killing of myeloma cells. So next slide. Going into sort of the three mechanisms, so the three types of drug more specifically. You see on the left what I just described. And as the bispecifics, they have an Fc region, which is what is leading to an extended half-life of these drugs so that they don't have to be administered very frequently. As I explained, the ADCs, they lead to an internalization of a chemo payload, so it does not require T cell engagement. It means that the immune effects that is part of the other two types of treatment is not relevant here, so it's not associated with CRS or neurotoxicity. What is a class effect of these payloads are ocular events. So that's quite common, but we know that this is attacking a part of the eye that is sort of continuously repleted. So this is reversible in practice all patients. So if you have this problem will go away if you stop the drug or dose it less frequently or in a lower dose. On the bottom, you see, if you look a bit on the right there, this is dosed every 3 weeks or every 4 weeks from the start of treatment. And I also have to comment that in Norway, we have started using these drugs, and we have on the national levels said that we will start these patients on every 8-week cycle in both combinations to avoid getting this ocular problem in the beginning. And in the studies, we see that that's where patients usually end up, 8 weeks or more as an interval between them. between the doses. Besides what we do with dose adaptation, we start at 8 weeks. The formal indication is every 3 or 4 weeks. To reduce frequency and to reduce dose are the main ways we manage these side effects. And it's also important to use artificial tears continuously. Yeah, I think you can go to the next slides where you look at the bispecifics. As I mentioned, they target both T cells and BCMA on the myeloma cells. They also have an Fc portion that leads to long half-life. Here you see quite frequent this CRS. That means that there's a large immune reaction when these drugs start to work, usually within a day or two. To mitigate that, we use step-up dosing. We give a smaller dose first. We do subcutaneous administration for most of these drugs. And sometimes we use prophylactic tocilizumab to prevent that. These patients become at risk of infections to a larger degree than the previous drugs we discussed. So it's common to use primary immunoglobulin substitution even often before we start the drug, because all of these patients will need that. They also get antibacterial prophylaxis with Bactrim, and in some places even broader antibiotics. These are usually started weekly and then going to bi-weekly and monthly. In Norway, we go from in all these drugs to monthly dosing from the 3rd cycle. So we try to reduce this quite quickly because we don't think the efficacy is less, and we have lesser problems with infections and CRS. So next slide. The CAR-Ts, I explained them before. We have 2, ide-cel and cilta-cel, in addition to the T-cell activation part. They also have this extra molecule that is co-stimulatory, that is a really strong activation of these T cells. There are some problems with these drugs. They have neurotoxicity, Parkinsonism, they have gastrointestinal toxicity, and they have CRS. The CRS comes a bit later, especially for cilta-cel, where the CRS comes after 5 to 7 days, a bit earlier for ide-cel after a couple of days. And we see the CRS in the beginning of this, or after this dose has been given. The very different thing about this drug versus the others is that it's only one infusion. So you get one treatment, and then the CAR T cells work quite fast, and also persistence is important to keep the efficacy. But we don't actually know how long that is important because we get a lot of reduction even after the first dose. So some dangerous side effects, but very efficacious treatment. So next slide. Here's an overview over the efficacy, and they are not in the same sort of situation. If you look at in the middle of the bispecifics, that's late-line disease. So they have a response rate of about 60-70%, and a PFS of about 1 to 1.5 years, usually. If you look on the right, ide-cel and cilta-cel, they have randomized trials, cilta-cel in 2nd line, ide-cel in 3rd line. You see the response rates are higher with cilta-cel than with ide-cel. That's partly because of earlier line, maybe also because cilta-cel seems a little bit more efficacious. And then PFS in cilta-cel is not reached, and the control arm is sort of as expected, 12 months. You look at ide-cel, you see the control arm of 4.4 months. This is on the bottom, but it means that this is a very difficult patient population to treat with a PFS of 13 months for ide-cel. Looking on the left for Belantamab with the 2 randomized control studies in 2nd line, you see on PFS on the bottom, you see a very large increase in PFS of a tripling in DREAMM-7, and we don't really know yet for DREAMM-8, but definitely much better. Also on the response rates, the CR especially, you see a big difference between Belantamab treatments and control arms. So next slide. Looking at safety, they are very different. CRS and ICANS are only there for bispecifics and CAR-T, not for ADCs. Neutropenia is a common problem in all treatments with myeloma, but not very problematic. I think especially important are the infections. I think these numbers is a little bit difficult to interpret, because my apprehension of this is that this is a really big problem with bispecifics for many months and years, as long as they are on the drug. For CAR-T, it's mostly a problem the first months, maybe half year. For bispecifics, it's all the time they are on the drug. For ADCs, I would say the infection problem is quite small. I think this is better illustrated in the text below the table, where you see quite large single center study showing grade 3 infections with ADCs for 8%, bispecifics 40%, and CAR-T 26%. That is more sort of how I feel it is in the clinic. And on the bottom, you see ocular toxicities only with ADCs, CRS and neurotoxicity only with bispecifics and CAR-T, while infections is the biggest problem in the bispecifics. Next slide. So key takeaways, BCMA-directed therapies, we have many of them now, but they are very, very different. And we don't have any comparative studies, but there are pros and cons of all. ADCs, very tolerable, can be given to almost everyone, I would say. Off the shelf, available, not frequent dosing, usually give every 8 weeks or even less. Bispecifics, quite exhausting, I would say, for patients and doctors when they're constantly on this, because there's lots of infections and virus infections and so on. But very efficacious drugs. We haven't seen recently, but not approved yet, also data for bispecific in second line, which looks really impressive. And CAR-Ts, extremely efficacious treatments, especially cilta-cel, and the earlier lines you give it, the better. But they have serious side effects that are important to know about.